Very good evening, everyone. Welcome to day 6 session of CUET PG Prarambh Series. Please confirm in the chat box if my voice is loud and clear and is the screen visible. Good evening, Vidya. Good evening, Vinod. How are you all? I'm sure you all are enjoying the CUET PG Prarambh Series classes. Okay, thank you for confirming, Kailash and Vidya. Yeah, very good evening, Neelam. So I can see some more are joining. So I'll just start the class in a minute. Yeah, very good evening, Janvi. Very good evening, GCM014. So today in this session, you will know about recombinant DNA technology and principles of gene cloning. So here in this session, you will know what is recombinant DNA technology, what is gene cloning, how it is done. We'll talk about tools of recombinant DNA technology in detail and also its applications. So tell me, before starting with this session, I would like to know how many of you have actually performed gene cloning? Like nowadays, kits are available in the laboratory. So I would like to know how many of you have performed gene cloning. Have you ever prepared, have you ever constructed a recombinant DNA molecule? Yes, is there anyone? Yes or no? You can please, you can write in chat box yes or no. Because I'm sure you know that this technique is very, very important, right? So, how many of you have performed or have seen actually? If not performed, have you ever seen in your colleges, in your universities? Yes, Drishya? Okay, that's great. What about others? Vidya, Neelam, Janvi, Shramalina, Kailash? So, why we are studying about this topic today? Yes? So obviously, one first reason you all know that it's in your syllabus, right? For the exam, you have to prepare this topic, right? But what is the another reason? We all belongs to life science, right? And there should be always there is always something new to learn, right? And this is a very important technique that being a life science student, we all need to know about it, right? Okay, so I can see some of you already know about it, but majority says that they don't know about it. So I'm sure that this session would be very interesting for you because if you don't know anything about any technique, so basically you will be learning it from the scratch, right? And those who already know about it, it will be a revision for you all, right? So let's study it in a very interactive way. Okay, it's not that it should not be one sided. You all are welcome to post your suggestions. If you have any question, please post it in the chat box. So let's get started. So let's first understand what is recombinant DNA technology, right? Recombinant, you can see a term here. So basically, here, this technology, it's all about joining DNA molecules from different organisms. See, this particular statement, it's very important that what you are doing here, you are joining the DNA molecules. That is two DNA molecules you are joining. But these two DNA molecules are not from the same organism, okay? They are from different organism. Now, question arises, what are these two DNA molecules? So please remember that first DNA molecule will be the DNA fragment or the DNA which will be carrying your gene of interest, right? What will be your gene of interest now? So let's say that you observe a plant which is showing herbicide resistance. Okay. Or let's say there is a bacteria which we are trying to grow in a lab and it is growing in presence of antibacterial agents also. So definitely, if a particular organism is showing some characteristic, that means the reason is that it has a gene for it, right? If a bacteria is showing antibiotic resistance, that means definitely it must be having a gene for it. Because we all know that whatever characteristics we see in an organism, it's all because of the genetic makeup of that organism, right? If, like, even if we take our example, if we have anyone is having a blue eyes or black eyes, or having let's say long hairs, right, or somebody is bald, so definitely there is a reason for it. All these characteristics are because of the genetic makeup of an organism, right? So similarly, if we relate it to the bacteria, if we relate it to the plants, right, some plants are tall, some are dwarf, right, some bear blue flowers, some bear white flowers, some bear red flowers. Similarly, some bacteria survive in presence of antibacterial agents. Some bacteria can survive in extreme temperature, right? So definitely it possesses genes which will allowing it to survive in some extreme environmental conditions, right? Or will possess a particular trait because it has a gene for it. So here we are using one of the DNA fragment which is having a gene of interest. Okay, that is, this is the trait we want to introduce in other organisms as well. So obviously we'll take this gene of our interest from the organism to which it already belongs. So first you will be isolating the DNA fragment carrying gene of interest, and second DNA molecule will be the organism which can provide, like, which second DNA molecule will be which will be able to carry our gene of interest into the organism in which we want to show the trait. So let's understand it with an example. So here you can see this is one organism which is possessing the gene for a particular trait. So this gene, we will be getting the gene of our interest, we will be isolating it from one organism, right? So obviously we have to isolate the DNA. Now we are not interested in using the entire DNA. We are only interested in the fragment, okay, or I should say the gene of our interest. So basically, we won't be using the entire DNA molecule. We have to break it into small fragments, and the fragment which will be carrying a gene of interest we want to use. And the another DNA molecule that I am talking about will be a vector. Okay, or you also call it as a vehicle, because it will be carrying our gene of interest and it will introduce it into the host organism where we want our gene to get expressed. So here you can see in this diagram that the another DNA molecule, the vector that we have used, is nothing but a plasmid. And I'm sure you all know what a plasmid is. If you don't know, then you can please note it down. That plasmid is a cell, like it's a extra chromosomal DNA material present in a bacteria. Like here you can see, this is like, if you see the structure of the bacteria, it has a cell wall, cell membrane, chromosomal DNA, and other than that, like it has the chromosomal DNA, and other than that it will have the extra chromosomal material which we called as plasmids. They are self-replicating extra chromosomal DNA material, and they have the genes which provide and which allow the bacteria to survive in unfavorable environmental conditions, like maybe in the presence of antibacterial agents or in different environmental conditions. So from one organism, you isolate the gene of interest. From another organism, you isolate the plasmid, which will be acting as a vector here. Now, other than plasmid, you can use cosmid also, lambda phages also. Now, what are cosmid, what are lambda phages? I'll talk about that later. We'll go step by step. Right now, we are trying to understand what is recombinant DNA technology. So will be joining the DNA molecule 1, which will be carrying a gene of interest, to the vector. Now you can see that there is some space in this vector, right? How we got this space and how we got these fragments. So there are certain enzymes that we use in recombinant DNA technology The very popular enzymes are restriction enzymes, which breaks the DNA into small fragments. Okay, they recognize a specific sequence and cleave it from there, resulting into breaking of the DNA into small fragments. Similarly, we can open the vector. Like let's say there is some sequence here which will be recognized by a particular restriction enzyme. So enzyme will cut from here and resulting it into opening of the vector. And now here we can introduce our gene of interest. So again, for joining our gene of interest into the vector, we need another enzyme called as ligase, which will act as a glue and join these DNA, both these DNA molecules. So the structure that is this DNA molecule that is constructed is called as recombinant DNA molecule. We also, this recombinant DNA molecules are sometimes also called as chimeric DNA because they can be made of material from two different species. And once this recombinant DNA molecule is formed, it is introduced into host organism. What is the host organism? What is your host organism? The organism in which you will be introducing your recombinant DNA molecule. It can be Escherichia coli, simple prokaryotic organism, or it can be yeast or plant or any other cell. And once the recombinant DNA molecule has been introduced, it will form its copies. The gene present will be transcribed. Later on, the messenger RNA form will be translated and form a functional product. So question arises, why you are preparing this recombinant DNA molecule? Why we are making these new genetic combinations? We are making these new genetic combinations because these are very much valued to science, medicine, agriculture, and industry. We can develop recombinant insulin. We can develop many more things are there, right? Growth hormones, recombinant growth hormones we can develop. Similarly, we can develop plants with improved yield, right? Pesticide resistant plants, insecticide resistant plants we can develop. And even we can use this technique in food industry, in biotech industry, in pharmaceutical industry to produce the products which are very much useful for the humans. And this particular technique, it was developed in year 1973 by Boyer and Cohen. These two scientists, they developed recombinant DNA technology, showing that genetically engineered DNA molecules may be cloned in foreign cells. Yes, you are absolutely right, Shivani, that extra chromosomal genetic material, it's the plasmid. And yes, transgenic plants, correct, Shivani, medicines, insulin, all that we can develop using recombinant DNA technology. Students, I would like to know, are you all comfortable with the English language or you want me to speak in Hindi as well? All the participants, to all the participants I'm asking, are you all comfortable in English or you want me to speak in Hindi as well? आप सबको समझ में आ रहा है English में? Is there any candidate, any participant who want me to speak in Hindi as well? Please quickly confirm. Okay, comfortable with English. See, I'm not able to see you. You all have to respond. You have to write in chat box yes or no. Okay, that's great. I'm really glad to see your responses. So majority, most of the students are saying English only. Okay, okay. Thank you for confirming, everyone. So yes, these are the recombinant DNA products I was talking about. So here you can see we can develop blood proteins, right? Factors 7, 8, tissue plasminogen activator. We can develop human hormones, right? Immune modulators, even vaccines as well. So many recombinant DNA products have been formed using this recombinant DNA technology. So this was the first term so far, like this was the first term we have discussed so far, right? Recombinant DNA technology. Now, another term is gene cloning. What is a clone? Can you tell me what is a clone? Like if we talk with respect to cells, right? Clone is what? They are all identical, right? A keyword is identical here. That it's a collection of molecules or cells which are all identical to original molecule or cell. See, participants, whenever you are studying, okay, whenever you are studying any topic, try to pick any keyword from there. It's not that you have to learn the entire definition. Like here, you can pick a word identical. That will be your keyword, okay? Clear to all? So you can see identical is the appropriate term here. So what is a clone? It's a collection of molecular cells which are all identical to original molecular cell. Like for example, when the bacteria divides through binary fission, right? What happens? It produces the two daughter cells which are identical to the parent cell, right? So we can say that they are clones. So always remember, clone is a collection of molecular cells all identical to original molecular cell. So how do we define gene cloning? Gene cloning can be defined as insertion of a fragment of DNA carrying a gene into a cloning vector and subsequent propagation of recombinant DNA molecule into host organism. So you can say it's a process of manipulating DNA to produce multiple copies of a single gene or DNA fragment can be formed. So basically the diagram that I showed you, right? It's, if you go through the entire process, it is the gene cloning that we are doing. But you can see that in this process of cloning, the very first step that we are doing is construction of recombinant DNA molecule, right? So you can say that how gene cloning is done, it utilizes recombinant DNA technology. I have come across, like, usually many students ask this question, like they always get confused. What is the difference between the gene cloning and recombinant DNA technology or genetic engineering? So please note that this recombinant DNA technology is also known as genetic engineering. But when you compare gene cloning and recombinant DNA technology, always remember that very first step in gene cloning is, like, it utilizes recombinant DNA technology. That is, you have to construct a recombinant DNA molecule first. And dear participants, through this session, I would like to really emphasize, okay, that whenever you study any topic, please try to study in this way only. Okay, always go step by step, because here you have to understand the concept. Because when you actually, you will give the exam, right? The exam, you know, this CUET PG paper will be MCQ based. So if your concepts are not clear, you won't be able to solve the questions, right? So here at Biotechnika specifically, we emphasize on starting from basic concepts, and then you can move on to the applied part, okay, or the complex information regarding that topic. So not only through this session you can remain connected with us, but you can also you can also join us, like you can also join Biotechnika Telegram group, or also you can download the Biotechnika app, where you will get to know about the podcast, the recent webinars which will be in the future, which will be conducted, about the certification courses, right? And also about the classes which will be going on related to the topic you want to study. So coming to basic steps in gene cloning now. So now we are familiar with two terms. I hope it is very much clear what is gene cloning and what is recombinant DNA technology. So here you can see the basic steps which are involved in gene cloning. So how you are going to learn it? You can see that there are basic 5 steps, right? So very first thing you have to learn is that there are total 5 steps. And the point that you have to emphasize here is the sequence of the steps, right? So here the very first step is that you have to construct a recombinant DNA molecule. So you will be taking a fragment of DNA containing the gene to be cloned, and then you will be inserting it into the vector, right? So let's say you want to introduce a antibiotic resistant gene into an organism. So first, from the organism where already this gene is present, okay, the donor which will be carrying the gene of our interest, from there you will be isolating the DNA, and then you will be breaking it with the help of restriction enzyme, you will be breaking it into small fragments. And then specifically the DNA fragment carrying the gene of interest, you will be introducing into another DNA molecule, which will be acting as a vector. It is also called as vehicle because it will be carrying our gene of interest. It's very simple, right? If you have to go from one place to another, like if you have a luggage, how do you travel? You need some vehicle, right? You need a bus, you need a car, you need a taxi, you need a bike, right? Similarly, this gene cannot move on its own, right? This gene cannot move on its own. It needs something which can carry it. And who will going to carry it? The vector will going to carry it. And who will be the vector here? The vector will be it can be a plasmid, it can be any artificial chromosome that we can create, right? Or cosmid, or any bacteriophage. So when we join the DNA fragment carrying a gene of interest with the vector, now vector also you have to open it up with the use of restriction enzyme, and then with the help of enzyme called as ligase, we will be joining these two DNA molecule. And result what we got? Recombinant DNA molecule. Now, next step is that we have to introduce this recombinant DNA molecule into host organism. Please note everyone that usually it's the Escherichia coli bacteria which is most commonly being used in the laboratory as a host organism. And obviously the host organism that we are using, why we are calling it as host? Because it harbors foreign molecule. Okay, it harbors foreign molecule, that is our recombinant DNA. Why we are calling it as foreign? Because for this bacteria, this is a foreign molecule, right? And please note, whichever bacteria you are using as a host organism, it should not contain any plasmid. The strain of bacteria that you will be using, like Escherichia coli, if you are using, the strain of Escherichia coli only that you have to use is the one lacking the plasmid. And this host organism should also be non-pathogenic. It should be non-pathogenic, easy to cultivate, right? So not only Escherichia coli, not only bacteria, you can use yeast also, we can use animal cell, plant cell also, right? But Escherichia coli, strain of Escherichia coli is most commonly used in the laboratory. So we'll be transporting it into the host cell. Now, what is the name of the process? Can you tell me? Have you heard of any process using which we can introduce the recombinant DNA molecule into the host cell? Maniraj, you have asked a very good question. What is restriction enzyme? I'll explain about that in detail later, but for you, I can just simply repeat that restriction enzymes are basically the endonuclease, the enzymes which can cut the DNA molecule. Now, how they cut, where they cut, exactly what these are, right? What are the other names we use to define restriction enzyme? I'll explain later, okay? I'll go step by step. So quickly, everyone tell me what is the name of the process that we use to introduce the recombinant DNA molecule into host cell? Have you heard of transformation? Have you heard this process, transformation? It's a natural process, right? Natural process of have you heard about horizontal gene transfer? Shrivani, yes, this is recombinant DNA technology, but what is the process we use in this gene cloning to introduce our recombinant DNA molecule into host cell? Yes, Shrivani, transformation. Yes, Jaya, absolutely right. Jaya Sai Ramya, very good. Okay, Rohita, you have not heard. Fine. Transfection. Okay, yes. So, see, basically, if we talk about transfer of a genetic material, right? Now, let's try to understand in this way. Take our example, okay, humans. How do we get the genes? We get the genes from our parents, right? So we have two processes, vertical gene transfer and horizontal gene transfer. Vertical gene transfer is a process where genes are transferred from parent to offspring. But horizontal gene transfer is a process where the genes are transferred, genetic material are transferred from one organism to another, which need not to be parent and offspring. Like transfer of the genetic material from one bacteria to another. So there are three natural processes. Natural process of horizontal gene transfer, Shrivan, you are right. Transduction, transformation, and conjugation. So transformation is a process where the genetic material, where this the DNA molecules are being taken up by the bacterial cells. Okay, like there are several plasmids or several DNA molecules in the environment. So bacterial cell can take up these materials, DNA molecules from the environment. So that process is called as transformation. So here, the introduction of any DNA molecule into any living cell is called as transformation. So it's the uptake of genetic material, usually in natural process if we talk about, it's the uptake of genetic material from environment by bacterial cell. And that natural process, even we are using in the laboratory, that we are introducing a recombinant DNA molecule into the living cell. So although this is a natural process, but even we can use artificial methods also, right? Like physical methods, chemical methods, biological methods we can use. Like one of you mentioned electroporation. Kailash, you mentioned electroporation, right? So that is a physical method using which we can introduce the recombinant DNA molecule into host cell. So I'll talk about that later. Again, as I always say, I'll go step by step. I'll not jump onto the another information. Let's complete it first, and then we'll move on to the transformation methods in detail later. So the vector transports the gene into host cell, which is usually a bacterium. However, we can use other type of living cells. Other types of living cells can be used. Now, what will happen within the living cell, within this host cell? This vector, the recombinant DNA molecule, will multiply. And you can see that it has produced identical copies, right? So obviously, our gene will also, the gene is also present, right? So numerous copies of genes are formed in turn, right? And even when this bacterial cell divide through binary fission, like as shown here, you can see that the copies of recombinant DNA molecule are passed to the progeny, and you can further, like we can say that replication can take place. And after a large number of cell divisions, like we can grow the bacterial cell in a media, right? So after a large number of cell division, a colony or a clone of identical host cell is produced. So each cell in the clone contains one or more copies of recombinant DNA molecule. The gene carried by recombinant molecule is now said to be cloned. So these are the basic steps in gene cloning. But I would like to ask you a question. Why is gene cloning so important? Yes? Can you tell me why gene cloning is so important? What do you think, that like why we are doing gene cloning? We can use other techniques also, right? Have you heard about PCR, polymerase chain reaction? What we do in PCR, we take a gene and we amplify it, right? PCR is based on natural process, the replication that takes place in living organism, right? So there also we can isolate, we can specifically amplify a gene. So why gene cloning is so important? Why you are studying about gene cloning still? Vivek, okay, for transferring desirable characteristics. Yes, you are right. At the end, even if we amplify the gene, we have to introduce into host organism, right? Vidya, to make multiple copies. Okay, so Vidya, that's what I'm saying. We can use PCR also, right, to amplify the gene. Don't you think we can amplify the gene? So even using PCR, Pratik, we can make lot of we can amplify the gene, right? Okay, Jaya Sai Ramya, you are saying to express gene of interest in order to get useful products. Yes, at the end you have to express the gene. Jaypreet, you are right, for expression of the gene. But specifically, I'm talking about see, When I say gene cloning here, I'm specifically talking with respect to the very first step that occurs that we do in gene cloning, that is construction of recombinant DNA molecule. So I am asking that is it so important to construct a recombinant DNA molecule? Don't you think we can simply isolate the gene of interest and we can amplify that gene, and then later on we can use like any physical method or or chemical method or biological method and introduce into organism? Why we are constructing recombinant DNA molecule? It's so lengthy process, right? It's not a one day process. If you compare it with PCR can be completed in few hours, whereas it takes weeks, if not months, to obtain a gene by cloning. Then why is gene cloning still used? Yes. So I'm getting lot of different answers. So yes, basically we want to introduce a new characteristics. We want to express our gene of interest into the organism, host organism, right? Into another organism where we want the trait. But please note it down that the importance of gene cloning is that we can actually obtain a pure sample of gene by cloning. Okay, like obviously as I mentioned earlier that we have a total, like we can isolate the total genomic DNA. You can break it into fragments, and then the fragment carrying your gene of interest you can introduce into a vector, and that vector will then be carry your gene of interest and will introduce into the host, will insert into the host cell, right? But I told you that even we can get a gene, pure sample of a gene by using polymerase chain reaction also, right? And PCR can be completed in few hours, whereas it takes weeks to obtain a gene by cloning. So why still we are performing gene cloning? So please note it down that obviously if you have to perform a PCR, right, you need to know the sequence of the gene first, because how do you design the primers? Yes, we already have the session conducted on PCR, right? So I'm sure you know what is PCR. The day one session, you remember, on PCR, electrophoresis, blotting. So obviously, if you have to amplify a gene, you have to use a primer, right? And in order for the primer to anneal to the correct position, the sequence of either side of the gene should be known, right? Either side of the gene of interest should be known. Annealing site, sequence of annealing site must be known. So it is easy to synthesize a primer with predetermined sequence. But if the sequences of the annealing sites are unknown, then the appropriate primers cannot be made. And this means that PCR cannot be used to isolate genes that have not been studied before. And this is the reason we have to do cloning. Second reason is that there is a limit to the length of DNA sequence that can be copied by PCR. 5 kb, 5 kilobase can be copied fairly, easily. But if you have a segment up to 40 kb, then for that you have to use specialized technique, right? If you talk about human genes, they are very large. So cloning is used if you want to have an intact version of long gene is required. So gene cloning is therefore the only way of isolating long genes or those that have never been studied before. Yes, is it interesting? Did you learn something new today? Tell me yes or no. Are you all ready to learn some new concepts as well? Please write, bring it on if you're ready. I want to see how much you are interested in learning new concepts. So the point is, dear participants, that try to compare and learn. Okay, when you have so many techniques to learn, if you compare and learn, you'll never forget, right? Thank you so much, everyone. So point is, try to compare whenever you see any two techniques you are learning and you come up with a concept, and you get confused. Okay, we are doing the same thing in cloning also, we can do it with PCR also, then what is the difference? Why still we use cloning if we have a PCR which is much faster? So the reason, these are the two reasons we are still using the cloning, right? Now, coming to tools of recombinant DNA technology. So today in this session, I'll be telling you about the 4 tools of recombinant DNA technology. These are the main tools. These are the which are the enzymes, vectors, host organism, and transformation methods. And even when we talk about enzymes, specifically the most important enzyme that you all need to know is restriction enzyme. But before again talking about restriction enzyme, you have to understand the name of the DNA manipulating enzyme, which are called as nucleases, because restriction enzyme is belong to these nuclease enzyme. So what are nucleases? Nucleases are the enzymes that degrade DNA molecule by breaking phosphodiester bond that link one nucleotide to next in a DNA strand. So basically, let's consider that, like I'm sure you all know that DNA is double stranded, right? So here, this string, let's say you consider it as a DNA, and these beads that you see, let's consider it to be a nucleotide, because DNA is a polymer and nucleotide is a monomeric unit of DNA. This nucleotide is a monomeric unit, right? So basically, this one nucleotide is attached to another nucleotide on the same strand with the help of phosphodiester bond, right? So DNA is double stranded. The two strands are there which run anti-parallel, right? So one nucleotide connected to another nucleotide on the same strand with the help of phosphodiester bond. However, the nucleotide, like nucleotide has 3 main components, right? In case of DNA, it has a deoxyribose sugar and it has a phosphate group, right? And nitrogenous bases. So nitrogenous bases, again, 2 types we have, purines and pyrimidine, right? ATGC. So if A is present here, it pairs with the T on the another strand with the 2 hydrogen bonds. If G is present here, it bonds with the C with 3 hydrogen bonds, right? So this bond that you see here, it's the hydrogen bond. You can see the labeling, okay? This is the hydrogen bond. So don't get confused between the bonds. Even you have to remember which bond is which one, okay? Phosphodiester bond is linking one nucleotide to another on the same strand. Now, what this nuclease enzyme will do, it degrade the DNA molecules by breaking phosphodiester bonds that link one nucleotide to the next in a DNA strand. So these nucleases are, we can say that nucleases are of 2 types, exonuclease and endonuclease. As you can see here, exonuclease are the enzymes which remove one at a time, which remove nucleotides one at a time from end of a DNA molecule. That is, they are breaking this terminal phosphodiester bond, right? And they are removing the nucleotides from the end. But when you talk about endonucleases, endo means within. So these are the enzymes which break the internal phosphodiester bond. So like here you can see it is breaking the internal phosphodiester bond, it is not removing the nucleotides from the end. So it is breaking the internal phosphodiester bond, right? So here the cleavage is taking place, and then further we have 2 fragments, DNA fragments, so further we can break them, right? And you can see at the end we have so many DNA fragments, right? So this exo and endo you have to remember. Now, what are restriction enzymes? Restriction enzyme belongs to larger class of enzyme called nucleases, or more specifically, you can note down that they are endonucleases, okay? Just now I told you nucleases are of 2 types, exo and endo. So restriction enzymes are endonucleases. And so you can also call them as restriction endonucleases. So what is the role of restriction enzyme? They allow the molecular biologists to cut DNA molecule in a very precise and reproducible manner required for gene cloning. See, let's take an example. You have a vector. You want to introduce your gene of interest here. So just imagine that if your vector, instead of being cut at a single position, like what we usually do, we cut the vector at single position and we open up the circle so that new DNA can be inserted here, right? Your gene of interest can be inserted here. Now just imagine your vector, instead of being cut at single position, it is being cut at two or more positions. So that results into generation of fragments, right? Your fragment, your vector itself being broken into two or more separate fragments, and then it will be of no use as a cloning vector, because we want the cloning vector to be open, the DNA molecule which we are using as a vector to open at single position, to be cleaved at a single position, open up in a circle, open up the circle so that new DNA can be inserted. And also, it has to be cut at exactly the same position, not random cleavage. Not, we don't want random cleavage. Similarly, this is your DNA fragment, we're carrying gene of interest. Now, if the enzyme that you're using cut the fragment from here, don't you think your gene of interest is disrupted? So that means gene cloning requires that DNA molecule should be cut in a very precise and reproducible fashion. So one of you asked me, ma'am, what is restriction enzyme? So restriction enzymes are the enzyme which cut the DNA molecule in a very precise and reproducible manner, which is required for gene cloning. Now, this raises a question: how this enzyme they are cutting so precisely? So these enzymes are cutting so precisely because they have a specific recognition sequence at which it cuts a DNA molecule. That is, like here you can see this is a recognition sequence. So enzyme will recognize it and will specifically cut the DNA molecule from here. It will not cut randomly. Random cut will not be there. And because these enzymes are they cut the DNA, we call them as molecular scissors as well. They are molecular scissors. We use scissors to cut the cloth or paper, right? So we are calling it as molecular scissors. They are popularly called as molecular scissors because we are cutting the DNA using them. So restriction enzymes are also called as molecular scissors or restriction endonucleases. And these are the enzymes which are naturally present in the organisms, especially prokaryotes, bacteria. Many enzymes has been isolated from the bacteria. So there they act as a natural defense mechanism. They don't allow bacteriophages to infect the bacterial cell. Bacteriophages, if they try to introduce their DNA into the bacterial cell produce these enzymes, they recognize the viral DNA and break it into fragments. So how these restriction enzyme cuts the viral DNA? Because they recognize specific sequences and cut the viral DNA. And Jaya Sai Ramya, you asked me, yes, with the help of restriction enzyme, okay, restriction endonucleases they are called as. And tools, Shrivani, the 4 tools are there: enzymes, vectors, host organism, and transformation methods. Okay. Now, Jaypreet, you are asking about Cas9. So Cas9 is actually related to CRISPR system, right? Now, here, it's a complete elaborate topic, right? So you can connect us through our Telegram group, or you can download the BioTechNika app, and in the chat there you can, like if you want, you can connect through various these groups, and you can post your question there, okay? Because if I try explaining you CRISPR-Cas9 system here, it will be a very elaborate, it's a very elaborate topic, okay? Let's not deviate from the topic right now. I hope you understand. So coming to classes of restriction enzymes now. Have you heard about classes of restriction enzymes? Shubham, you have asked a very interesting question, that why these enzymes do not cut bacterial genetic material. No, no, it's absolutely right. It's absolutely fine, Jaypreet. I really appreciate you, that I can see you are so eager to know about the enzymes and all, and I really appreciate you all for this. But we have a time limit, and as I told you, CRISPR-Cas9 itself is a very detailed topic, so that's why I won't be able to cover here. Right? Thank you for understanding. And Shubham, you asked me why these enzymes do not cut. So basically, the bacterial cell produce one enzyme, one of the class of enzyme called as restriction enzyme, and also they possess an enzyme that modifies the DNA. That is, they can produce methyl transferase. So basically, I told you that if the bacteriophage try to infect a bacterial cell, what is bacteriophage? The virus that infects the bacteria, virus that infects the bacteria, bacteria eating viruses are bacteriophages. So actually what they do, they attach to the bacterial cell, they introduce their genetic material in the bacterial cell. Now you know viruses, they are a bridge between living and non-living. They are non-living outside the host cell, right? They need a host cell to replicate. They uses the host cell machinery to replicate. So as soon as they introduce it in their DNA into the host cell, they use the host cell machinery to replicate, right? And they produce new phage particles and cause lysis of bacterial cell, that is killing of bacterial cell. So to protect itself from being killed by the bacteriophages, the bacteria develops a natural defense mechanism, that is restriction modification system. And this restriction modification system I'm talking about is where when we use the bacteria uses restriction enzyme and produce methyl transferase enzyme. So the restriction enzyme, actually they recognize specific sequence on viral DNA and specifically cut the viral DNA so into fragments. As a result, the viral DNA becomes inactivated and will no longer able to infect the bacteria. However, as Shubham asked, that why the bacterial DNA itself not get killed? Like let's say the restriction enzyme is recognizing this sequence GAATTC. There is a possibility this sequence is present in bacterial DNA also. So how the bacteria will recognize, how bacteria will know that its DNA should not be cut? So there is another enzyme called as methyl transferase, which modifies the bacterial DNA. Like if this sequence is present in bacterial DNA, it adds a methyl group to it. So when a methyl group is added, the restriction enzyme will not cut the bacterial DNA. I hope this answers your question, Shubham. Right? Now let's quickly move on to classes of restriction enzyme. You can see there are mainly three classes of restriction enzyme. Three different classes of restriction endonucleases are there, and type 1 and type 3 are actually a little bit complex, and they have a biological significance, but they have a limited role in genetic engineering. It's the type 2 endonucleases which are most commonly or majorly being used in gene cloning. So what is the difference between type 1, type 2 and type 3? Let's quickly just go through them, the basic difference. As these are enzymes, so you can see the structure, you can note about their structure, that type 1 are bifunctional enzyme. So I told you about two things about this restriction modification system of bacteria, right? So these are type 1 are bifunctional enzyme, that is they have both endonuclease and methylase activity. They will cut the DNA and they can add methyl groups as well. However, type 2 are unifunctional enzyme. That is, either they will perform endonuclease activity or methylase activity. Similarly, type 3 are also bifunctional enzyme. Then when we talk about subunits, type 1 have three different subunits. Type 2 has two identical subunits, and type 3 are having two different subunits. Now, these are enzymes, so definitely they'll be requiring some cofactors, right? So type 1 and type 3, they require ATP and magnesium ion. But type 2 is the only enzyme which does not require ATP. It requires only magnesium ion. And the very important differences in the recognition site. For type 1, the recognition site is bipartite and asymmetric. Bipartite means it's discontinuous. It's not continuous. Like for example, if you see this example, BCG1, CGA, NNN, TGC. So N can be any nucleotide. So basically, this enzyme recognizes CGA and TGC. So this N can be any nucleotide, and this is the recognition sequence. It's not continuous. So we are calling it as bipartite, and it's asymmetric as well. Asymmetric means that it does not have any symmetry. Like for example, if I write this sequence GAATTC. Now by default, we always write the recognition sequence for restriction enzyme 5' to 3'. Now as we know, DNA is double stranded. So here on the another strand, the sequence will be, you can see that the sequence is read same on both the strands, right? GAATTC. If we read it in 5' to 3' direction, GAATTC. Here also GAATTC, right? So they have there is a, you can say that they are palindromic. Okay, we call such sequences as palindromic. But asymmetric sequence will be what? Which are not palindromic. They don't have symmetry. Like if I read this sequence 5' to 3', it is GGATG. Now, if you write the sequence on another strand, it is CATCC. So this is not palindromic, right? They are not read same on both the directions, right? So this is asymmetric. However, type 2, they often recognize 4 to 6 base pair sequence and they are palindromic. And type 3 also, they recognize 5 to 7 base pair, but the sequence is asymmetric. Now, one thing is recognizing a sequence. Another point is, where do the enzyme cut? Is it cutting at the recognition site only or somewhere else? So type 1 and type 3, if you see, type 1 is non-specific. It cuts 1,000 base pair from the recognition site. So if the recognition site is present here, it will cut some 1,000 base pair away from this recognition site. However, type 2 will cut same or close to recognition site, as I showed you here. EcoR1 is a type 2 restriction enzyme. So it has this recognition sequence and it will cut within this sequence, right? And type 3 also, it will be cutting 24 to 26 base pair downstream of recognition site. And here are some of the examples of type 1, type 2, and type 3. Jaypreet, yes, for type 2, it's the palindromic sequence, right? So quickly here you can see some of the examples of restriction enzymes. And these are the organisms from where these restriction enzymes are isolated. So basically like around 2,500 of restriction enzymes have been detected at different organisms, and more than 300 has been used in the laboratory during gene cloning or recombinant DNA technology experiments. So these are the organisms from which these enzymes are isolated. These are the recognition sequence. Now here you can see that we are, now please, one more point I would like to tell you before talking about these enzymes in detail. Please note that from now whatever restriction enzyme we are talking about, they will all be type 2. Because just now I told you that type 2 is the enzymes which are most commonly being used in the laboratory because they are easy to work with. They recognize the palindromic sequences, right? They don't require ATP. So if you compare it with type 1 and type 3, which have a biological significance, but they are less used in genetic engineering, type 2 are more commonly being used. Now, talking about the nomenclature of the enzymes. So like, for example, EcoR1 is isolated from Escherichia coli. So basically, if you see this first 3 letters, which are written in italics, they abbreviate the genus and species name of the organism, right? Like E has been taken from the first letter of genus Escherichia, and CO has been taken from coli, first 2 letters of the species name. And the 4th letter R, in this case, it comes from the bacterial strain. And these numbers that you see, okay, they are written in Roman. Okay, they are Roman numerals are used, and these are being used to identify specific enzyme from the bacteria that contain multiple restriction enzyme. So Roman numeral indicate the order in which restriction enzyme were discovered in a particular strain. So like EcoR1 is the first enzyme that to be discovered in this organism, Escherichia coli, and specifically its strain that you have to keep in mind. Similarly, like if you talk about Hind3, right? So here Hind3, so H comes from Haemophilus, first letter of genus, and IN comes from species, first two letters of the species name, right? Then D is the strain and 3 is the Roman numeral that indicate the order in which the restriction enzyme discovered in particular or in this particular organism. Now you can see the end part also, right? Here I'm using a term blunt and sticky end. So what are these blunt and sticky end? So far we have studied that these enzymes, they cut. Okay. But please note that the cutting is also done in different manner. The cutting can either produce the blunt end or can produce a sticky end. So why it is important to study about these ends and what are these ends? So I'll answer the first question, that it is important to know about the cuts, the blunt end or sticky end, because it has an importance in design of a gene cloning experiment. The exact nature of the cut produced by restriction endonuclease is of considerable importance in the design of gene cloning experiment. Now, what is blunt and sticky end? So just see this image carefully. Here, N can be any nucleotide, and you can see these the nucleotides which are written in red. Okay, the bases which are written in red, they are the recognition sequence for enzyme ALU1. Now, this ALU1, it is cutting the DNA it is cutting the DNA at the same position in both the strands, right? So it is cutting the DNA strand, both the strands, they are cut at the same position, that is in middle of the recognition sequence, and in turn result into production of the ends called as blunt ends or flush ends. They don't have any overhang region. They don't have any single nucleotide, string of single nucleotides at the end. However, when you talk about sticky ends, so here let's take an example of EcoR1 enzyme, which cuts the two DNA strand not at the same position. The cleavage is staggered. That is, if enzyme is cutting in one, like here in this strand, if the enzyme is cutting at this place, on another strand it will be cutting 3 to 4 nucleotide away from the nucleotide it is cutting on the first strand. So like it will not cut from here. 3 or 4 nucleotide away from the first cut, away from the cut it is making at the first strand. So that results into the DNA fragments that have some overhang region. That is, here you can see some string of short single stranded nucleotides, right, at each end. So these type of ends are called as sticky ends or cohesive ends because they can stick back by transient base pairing. Right? A pairs with T, A can pairs with T, T can pairs with A, T can pairs with A. Shramalinar R in EcoR1 represents the strain. Okay, it's the bacterial strain designation. The fourth letter in the restriction enzyme name is the bacterial strain designation. Right? Okay. And Samantha, you asked me that endonucleases are used in genetic engineering, then methylase enzyme is not used. Samantha, we can use methylase enzyme if you already know that there are recognition sites for the restriction enzyme present in our gene of interest. Then in that case, we can treat that DNA fragment carrying a gene of interest with methylase enzyme as well. Okay, it's not that we don't use it, like specifically when we prepare the library, genomic library, right? And if there is any such enzyme using that enzyme, right, so it can be used. It's not that you cannot use it. Okay, now, so here you can see blunt end and sticky end. Now, have you heard about linkers and adapters? Yes, have you heard about what are linkers and adapters? Have you heard about them? Okay, Kailash, you asked me how to identify the desired gene of interest from DNA fragments. So, Kailash, desired gene of interest, either you can use a PCR, which will specifically amplify your gene of interest, right? So, once your recombinant DNA molecule is formed, there is a possibility that some errors would have occurred, right? Proper recombinant, desired recombinant DNA molecule is not formed. Then, in that case, you can use PCR technique. And selection process is also there. So I'll talk about the selection process. Okay, let me first come to the vectors and then I'll explain you about the selection process. Okay, okay, most of you have. So yes, tell me, have you ever heard about linkers and adapters? See, linkers are short pieces of double-stranded DNA of nucleotide sequence that you can synthesize in the laboratory. Okay, and what is the role of the linkers? Like, why we are using linkers? Because here you can see that if you have the DNA molecule, okay, let's say this is a DNA molecule, and if you want to ligate it to the vector, okay, so usually the sticky ends are preferred, right? Because what happens, even if you're using the ligase to join the DNA fragment to the vector, the ligase is unable to catch hold of the molecule to be ligated, because they don't have any they don't have any blunt ends, they don't have any kind of overhang region which can stick back by transient base pairing, right? So therefore ligation of complementary sticky end is much more efficient. So if you're cutting whichever restriction enzyme you are using for cutting the DNA fragment, similarly the same enzyme also you have to use to cut the vector, so that the ends which are generated, and obviously the enzyme that you'll be using will be the one which generates sticky ends, so it is easy for them to join because they can stick back by transient base pairing, right? So if you have a blunt end molecule as shown here, so we want to convert them into, like, we want to convert the blunt ends, like we want sticky ends to be there. So for that, what we can do now, this DNA molecule has a blunt end. So how you will convert into sticky end? So for that, what we are going to do is we'll attach a linker. So a linker is a short pieces of double-stranded DNA of no nucleotide sequence. Okay, so basically this linker, you can see it already contain a recognition site for restriction enzyme. So based on whatever recognition site of restriction enzyme is present, we call that linker according to that name only. Like this is BAMH1 linker. If the eco R1 recognition site is here, we will call it as eco R1 linker. Okay, so here this is the BAMH1 linker, and we'll attach this linker to the blunt end molecule with the help of DNA ligase. Now these linkers also, you can see they don't have, they have the blunt ends, right? But actually when you perform the restriction digestion with BAMH1, what will happen? BAMH1, you know, it generates sticky end. So as the linkers are attached at the end of the DNA molecule, when you perform the restriction digestion with BAMH1, you can see that sticky ends are generated. So DNA ligase can attach linkers to end of large blunt-ended DNA molecule, right? And then finally we can use the enzyme, specific enzyme for the linker that we have designed. And then when you perform the digestion with BAMH1, it cleaves the recognition sequences, right? So like one or more than one linker can also be attached, okay? So like here you can see a chain structure. So many linkers are attached. But once you perform the digestion with BAMH1, it keeps the chains at the recognition sequence, producing a large number of cleaved linkers, and the original DNA fragment will now carrying BAMH1 sticky ends. However, the disadvantage of using a linker is that if the recognition site for BAMH1 is also present within your DNA molecule, then it will result into that in your cleavage of the DNA molecule as well. So this is a drawback of linker, right? And then what are adapters? They are also short synthetic oligonucleotide, but they already have one sticky end. So this is the difference between linker and adapter. So compare and learn. But again, that disadvantage of adapter is that they could ligate to one another. So these are the two terms that you have to keep in mind. Now, quickly, let me tell you about DNA ligase as well. Students, I've already extended the lecture. It's above 8. Is it fine? Shall I continue? I hope you are enjoying the class. Please write BIO if you want to know more about the gene cloning and recombinant DNA technology, because it's a very detailed topic, right? We cannot simply learn it superficially. I hope you understand that. And even if you have gone through the papers, which the type of questions asked in the CUET PG, I hope you understand that we need this kind of knowledge, right? We need this kind of information. Yes, are you all ready? Shall I continue? I need response from all. Okay, thank you, Avishka. Thank you, Shubham. Thank you for confirming. What about others? Quickly write BIO if you want me to continue. Okay, that's great. I'm sure you are enjoying the class, right? Okay, thank you. Thank you, everyone, for confirming. Thank you so much. So yes, DNA ligase, coming to DNA ligase, what is DNA ligase? See, DNA ligase is the enzyme which joins the DNA molecule, right? So it has a utility in DNA replication process. If there are single strand breaks, the discontinuity, like here you can see phosphodiester bond is missing. So this kind of discontinuity arises in double stranded DNA molecule during DNA replication. So there you can use DNA ligase. And here in recombinant DNA technology, we use DNA ligase to join the two fragments of DNA molecule. And DNA ligase, basically, you can see that it helps in, like it joins one nucleotide to another with the help of phosphodiester bond. Now, let me quickly tell you about vectors also. What is a cloning vector? Vector, or also called as vehicle, right? So cloning vector is a DNA molecule which is capable of replicating in a bacterial cell. And it is used to amplify a single molecule of DNA into many copies. So as I told you earlier, that once the recombinant DNA molecule is introduced into the host cell, it replicates it from multiple copies, right? Now question arises, can you use any DNA molecule as a vector? I gave you the example of plasmid, but there are so many plasmids, right? Some plasmids are very smaller in size, some plasmids are very large in size, right? So can you use any DNA molecule as a vector? So answer is no. You cannot use any plasmid or any DNA molecule as a vector. There are certain criteria which has to be fulfilled. So you can use any DNA molecule as a vector only when it has a small size, that is less than 10 kb. Why we need less than 10 kb? Because it's easy to separate it from chromosomal DNA. Okay, then it should have origin of replication, because then only the plasmids or your vector will be able to replicate independently from host chromosome. It should have multiple cloning site, that is it should have recognition site for several restriction enzyme, and it should have selectable marker genes as well, that is which can have the marker genes, that means which can indicate that whether the transformation has taken place or the desired recombinant DNA molecule is present or not, right? So here you can see that we have different genes here, ampicillin resistance, tetracycline resistant gene, or the enzyme for beta galactosidase that can be used as a marker gene, right? Now coming to types of cloning vector. So here you can see we have a different types of cloning vector that can be used. So we have a plasmid, we have a cosmid, bacteriophage lambda, bacterial artificial chromosome, yeast artificial chromosome, just to name a few. Okay, a lot of cloning vectors are there. And I would quickly like to tell you about the plasmid, the one of the first vector that was developed was PBR322. So here the P, like if you see the nomenclature, the P stands for the plasmid. Okay, the P indicate that it is indeed a plasmid. BR actually it identified the laboratory in which the vector was originally constructed, like it represents the BR, that is B for Bolivar and R for Rodriguez. So these two scientists, they developed this vector. And 322. 322, it distinguishes this plasmid from other which are developed in the same laboratory. Now, why PBR 322? It is the most, like this particular vector is the most common vector that was being used initially. So why this vector was so popular? So there are three features that makes it most popular. One useful feature is size. So you can see it is only around 4.3 kb. And in the previous slide, I told you that the size should be less than 10 kb, right? Ideal size should be less than 10 kb. So yes, it fulfills that criteria. Then second feature was that it carries two set of antibiotic resistant genes. So basically, this antibiotic resistant gene is important because it helps in selection, right? So here you can see it has ampicillin resistant gene and tetracycline resistant gene is there. And even you can see it has a multiple cloning site present between these marker genes. And third advantage is that it has a high copy number. What is copy number? It refers to number of molecules of the individual plasmid that are normally found in a single bacterial cell. And also it has origin of replication. So it fulfills all the criteria, right? So it has a high copy number. And question arises, how we will be introducing the gene into this vector, our gene of interest into the vector. So either we can introduce a gene into this ampicillin resistant gene or tetracycline resistant gene. So here there is one term which we often use, is called as insertional inactivation. Okay, so basically this is a normal vector molecule where the gene are intact. Okay, like let's say we are talking about PBR322. So here we have, let's say this is tetracycline resistant gene. So it is intact. But if we introduce our gene of interest between this gene, tetracycline resistant gene, don't you think the gene will be inactivated? Because the gene sequence is no longer remains the same, right? And when the gene gets disrupted means it will not produce its product. No gene product will be there. So this is a normal vector molecule where the gene remains intact, but in recombinant vector molecule, the target gene is disrupted. So here you can see in case of PBR322, this is ampicillin resistant gene, this is tetracycline resistant gene having recognition site BAMH1 for BAMH1. Now, if I introduce this vector into the bacterial cell will survive if I grow it in presence of ampicillin and tetracycline, because it has the genes which provide resistance to the bacteria for these antibacterial agents, right? However, if I introduce the gene of interest into this tetracycline gene, so what I did, I performed the restriction digestion with BAMH1 enzyme and I introduced the new DNA here. Now, as the gene is being introduced, the gene of our interest is being introduced, what happens? The tetracycline gene is become inactivated. So we can now write that the recombinant PBR322 molecule, which carries an extra piece of DNA in BAMH1 site, is no longer conferred tetracycline resistance because it is now disrupted. The gene is now disrupted. So it will be resistant to ampicillin but sensitive to tetracycline. So here, now question arises, why we are introducing the gene into tetracycline, our gene of interest into tetracycline? Because I told you we are using antibiotic resistant genes as a marker. We are using the antibiotic resistant gene as a selection marker, right? Atanu, you asked me that what is the difference between marker and reporter gene? Okay, so basically, I'll answer this question in a while. Let me just complete it with. Let me just complete with this. Okay. Let me just complete how we perform the screening. So basically, here The tetracycline we are using as a selectable marker gene. So basically what happens, we can identify two things. We are using selectable marker genes, so we are using them to check for whether the transformation has taken place or not, and to check whether the desired recombinant DNA molecule has been introduced or not. Because participants, always remember, dear participants, that whenever you are using any technique, the chances of errors are always there. I mentioned earlier that there are 5 major steps in gene cloning, where first major step is construction of recombinant DNA molecule. So there is a possibility that instead of the vector carrying a gene of interest, it self-ligate to itself, right? There is a possibility that unligated vector remains, or unligated DNA fragments remains, or there is a possibility that instead of a gene of interest, some non-desired gene has been introduced into the vector, right? So these are all the possibilities that could have happened. We want only the vector with a desired recombinant DNA molecule, desired gene of interest, right? So how do we check for that whether the bacterial cell which is transformed carries the desired recombinant DNA molecule only? It does not carry self-ligated vector, right? Or on the very first place, we have to check whether transformation has taken place or not. So all these things we have to check, right? So for that, we perform screening. So here you can see on the screen the three, like you can see three possibilities. Like first is indicating that no transformation has taken place. So as I mentioned earlier, that we will be using the host organism, the bacterial strain, which will not having, which will the strain, the one which is not having any plasmid, right? So that we can check for transformation. So no plasmid. So obviously this bacterial cell, if it lacks the plasmid PBR322, it will be ampicillin sensitive and tetracycline sensitive. However, another cell is representing the vector which is self-ligated, okay, intact plasmid. That means it is the normal vector molecule, not the recombinant one. However, the third one represents the recombinant one. So first we'll check for transformation. So what we're going to do is we'll grow the cells into a media containing ampicillin. So obviously those cells will only survive which will be having a vector, because PBR322 is having this normal vector molecule is having an antibiotic resistant gene. If no transformation has taken place, how this bacterial cell will survive, right? So only transformants will produce colonies. And once we have checked for transformation, next we will perform the we'll check for tetracycline resistance or sensitivity. That is, now we'll check for desired recombinant DNA molecule. So first one will no longer able to grow. So that means in this plate, either we'll have the intact plasmid or clone plasmid. So to further confirm that our bacterial cells carry the recombinant plasmid and not the intact one, we'll now perform the replica plating. We'll make a replica of this plate and we'll grow it in presence of tetracycline. So obviously the non-recombinants will only survive because they will have the tetracycline resistant gene, right? But our desired, a recombinant vector molecule will not survive because it is tetracycline sensitive. So from the master plate, we know where, like here we can see that these two colonies are missing if we compare it with master plate. So further we can take up we can pick a cells and we can grow it in fresh media. So this is how we perform the screening for PBR322 recombinants. So here you can see that there are a lot of vectors which can be used in the gene cloning, and we can use plasmids, we can use bacteriophages, right? Lambda and M13 are most common bacteriophages that are used. We can use cosmids, which contain the cos site of bacteriophage lambda and plasmid. So it's a hybrid, the plasmid containing bacteriophage lambda cos site. Then you can use artificial chromosomes also, right? So for if you want to introduce, if you are using the host cell yeast, then for that you can use these artificial chromosome, right? And you can see that all these vector, they differ in their size limit, less than 10 kb, some has 5 to 20 kb limit, okay? So always remember that the vector that you'll be choosing, okay, the vector that you will be choosing, it will be you will be choosing it on the basis of two things. First, the host organism in which you want to introduce your recombinant DNA molecule. Because if you are using a eukaryotic cell, right, if you want to express a gene, eukaryotic gene, then definitely you have to use a organism where that gene can be expressed, right? So even if you are using a eukaryotic cell, so accordingly you have to select a vector. And second, the insert capacity. Depending upon what is the size of your gene of interest, accordingly you'll choose the vector. And Jaya Sai Ramya, you asked me what is the difference between cloning and expression vector. So Jaya Sai Ramya, as the name indicates, expression vector, right? Expression vector is, as I just now, just as I mentioned, that your vector you are choosing, it should be such that when you're introducing the gene and that will be introduced into the host cell, that gene should be expressed. So expression vector is being used when is the vector which carries some regulatory elements which allow your gene of interest to express in the host organism. Mainly three regulatory elements are used: promoter, terminator, and ribosomal binding site. So these are the vectors, expression vectors, which allow your gene to be transcribed and translated. Okay. And Atanu, you asked me that cloning vectors are mainly used to increase the number of target gene. Meanwhile, expression vectors are mainly used for expressing. Yes, you are right. Expression vectors, if you have to simply make the multiple copies, you can use cloning vector, Jaya Sai Ramya. But specific your aim is that you want to express your gene. So for that, definitely you need a regulatory elements. And for that, the expression vector can be used. Now, coming to host organism quickly. So as I mentioned earlier as well, that host organism, Escherichia coli, is the most commonly used host in recombinant DNA technology. However, other than this, you can use other organisms as well, right? And whatever strain of Escherichia coli you are using, it should be non-pathogenic. And usually this is the preferred host for protein production due to its rapid growth and ability to express proteins at very high levels. And then transformation methods. So transformation method, in the beginning only I told you it's a process by which an organism acquire exogenous DNA. It can be a natural process or artificial process. So natural process, I told you, it's a horizontal gene transfer, right? It is one of the process of horizontal gene transfer which occur naturally. And artificially, we can perform transformation by using chemicals like calcium phosphate. You can use physical methods like electroporation, microinjection. Biological method, we can use vectors also, uh, viral viruses as a vectors also, right? And please note that it's not that whichever bacteria, like it's not easy that whatever strain of bacteria you are using, the bacteria will take up the DNA. No. If you are using Escherichia coli, then also the Escherichia coli can take up only limited amount of DNA under normal circumstances. So it's very important that we transform these species efficiently. So the bacteria has to undergo some sort of physical and chemical treatment which can enhance their ability to take up DNA. And cells that have undergone this treatment, we call them as competent. So here you can see how we can make a competent cell. So you can soak the bacteria like Escherichia coli in ice cold salt solution. So here calcium chloride is being given. So like 50 millimolar calcium chloride can be taken. So it is traditionally used. However, other salts like rubidium chloride can also be used. And basically, this calcium chloride will allow the DNA to precipitate outside of the walls, or perhaps it is responsible for some kind of changes in the cell wall that improves DNA binding. But please note that even after calcium chloride treatment, this exogenous DNA is still not been taken up by the cell. So further, you have to perform a heat shock treatment that you will be once the DNA has been added to the treated cell, it remains attached to cell exterior. So further, for the movement of the DNA into competent cell, you have to stimulate it by raising the temperature to 42 degree, and finally it will be taken up by the cell. And lastly, coming to the applications. So in the past century, recombinant DNA technology was just an imagination that desirable characteristics can be improved in living bodies by controlling the expressions of target genes. However, in recent era, this field has demonstrated unique impacts in bringing advancement in human life. So this particular technique has multidisciplinary applications and potential to deal with important aspects of life. For instance, you can improve the health, right? Enhancing food resources, resistance to diverse adverse environmental effects, like in particularly in agriculture. We can develop genetically modified plants which can be resistant to harmful agents, enhanced product yield, right? Adaptability, they can show adaptability for better survival. Then we can also use them in forensic science, energy applications, archaeology as well, right? So a lot of applications are there. So with this, I have come to the end of the session. Let's quickly have a concept check. So tell me, which is the most popular and widely used engineered plasmid vector? Is it PUC8? Is it PUC vectors like PUC8, or is it PBR322? Is it PSC101, or it is PUC19? Quickly answer this question. Can I have the answers, please? PBR322, sure? Yes, very good. Very good. Please note PSC101 is the first cloning vector which was used in 1973 by Boyer and Cohen. So SC that you see here, it stands for Stanley Cohen. Okay, but PBR322 is the first widely used cloning vector. Okay, and PUC8 are derived from PBR322. Then can you tell me which type of restriction endonuclease does not require ATP? What will be the answer here? Option 2. Very good. Type 2. Correct. Then moving on to next question. Very good. Very good. I'm really glad to see all the answers coming. Thank you so much. Moving on to next question. Which one of the following cloning vector incorporate highest length DNA fragment? Is it bacterial artificial chromosome? Is it yeast artificial chromosome? Is it phage mid, or is it cosmid? Yak? Yes, absolutely right. It is yak. Correct. Yeast artificial chromosome. Then which one of the following statement is not true for competent cells? Yes? Which of the following statement is not true for competent cell? They give better transformation efficiency during cloning. They could be produced by calcium chloride treatment. They possess high copy number plasmid, or heat shock assist in increased transformation. Not true, you have to answer. See, we are making competent cells so that we can get better transformation efficiency. So this is true. They could be produced by calcium chloride treatment. Yes, we have given calcium chloride treatment, and even we have used heat shock to increase the transformation, right? So yes, it will be 3. Absolutely right. Very good. And I think this is the easiest one. Which one of the following could not be utilized as vector? So see, students, this kind of questions you can expect. Dear participants, you can expect these questions in the exam. Okay. So obviously, which one could not be utilized? Plasmid, you can utilize. Bacteriophage, what about bacteria? What about phage mid? Bacteriophage lambda, bacteriophage 13 we can use, right? Pratik, you are right. It will be bacteria. Yes, again, I'm getting answers from all. Very good. Correct answer. Very good. So that means you have understood the concepts clearly, and I'm glad that you are able to answer all the questions correctly. So with this, I have come to the end of the session. Thank you for your time, and I'm sure that you have learned something new today and you have enjoyed the class. We always here at Biotechnika try to, we want you all to learn something new. And especially those students who have, the participants who have attended the session till end, you will be getting the notes. So the link has been shared. You can download the link from there. And Atanu, I'll just answer your question. You asked me what is the difference between marker gene and reporter gene, right? Marker gene, it is used to determine if nucleic acid sequence, like a recombinant DNA molecule, has successfully inserted into organism's DNA, right? We can use antibiotic resistant gene or any beta galactosidase gene. But reporter gene is a gene that researchers, they can attach to a regulatory sequence of another gene of interest in cell culture, animals or plants. Okay, so they are chosen as reporters because the characteristics they confer on organism express them as easily identified and measured. Okay, so thank you everyone, and all the very best with your preparation.